Friends, welcome to our YouTube channel. Friends, in this video, we're going to see most frequently asked quality assurance interview questions. So let us start with the first question. Define quality assurance. Friends, quality assurance is a broad range of concepts uh, that contain all matters which individually or collectively that are going to affect the quality of the product. So the QA is mainly concerned with the quality of the product, and QA is mainly concentrated on planning and documenting the procedures to assure quality. So what is documentation? Friends, documentation is a written procedures, records and instructions, quality control tests with procedures involved in all manufacturing activities of the drug products. So documentation means the records, instructions, quality control tests with procedures involved in all manufacturing activities of drug products. So coming to the next question. So during in-process Q.A checks, what needs to be checked? Friends, the following needs to be checked: environmental monitoring, measured values from the process, equipment, and the measured values obtained from the person's process attributes like weight, hardness, and stability. So QH in-process checks include environmental monitoring, measured values obtained from the process equipment like the temperature, RPM, and the measured values obtained from the person's timings and entries. So what are in-process checks? Friends, in-process checks are the checks which are carried out during an activity to monitor and where necessary to adapt to the process to ensure the product conforms to its specifications. So what needs to be checked during air handling unit validation? Friends, during the air handling unit validation, the following tests shall be carried out, which include air velocity and number of air changes, the airflow pattern, differential pressure, temperature and relative humidity, static area qualification, dynamic condition qualification, non-viable count, the microbial count, area recovery and power failure study. These 8 things need to be tested. So what precautions should be taken while collecting in-process sample? Friends, while collecting in-process samples, you must prevent the sample from contamination, so that do not collect the samples with bare hands. Use gloves while sampling. So what are the factors which influence the tablet hardness? The force applied for compression. The important factor is binder quantity. More the binder, more is the hardness and presence of moisture in the granules. So if the leak test fails during in-process checks, what need to be done? So immediately stop the packing process and check for the sealing temperature, check knurling quality and change in the printed foil, and check and quarantine the isolated quantity of packed goods from the last passed in process. Collect random samples and need to perform a retest. Blisters from the leak test passed containers shall allow going further, and rest must be blistered or defoiled accordingly. Friends, what is 21 CFR Part 11? Friends, the title CFR means Code of Federal Regulation, and which deals with the Part 11 deals with Food and Drug Administration guidelines on the electronic records and electronic signatures in the United States. So what is change control? Friends, change control is a general term which describes the process of managing how the changes are introduced into a controlled system into the validation. This implies how changes to the validated system are made. Change control is needed when to demonstrate to the regulatory authorities that the validated system, whatever the validated system is, remain under control after the changes are made. So what is SOP? Friends, this is the most commonly asked interview and basic questions. The SOP means Standard Operating Procedure, which is a document which describes the operations to be performed in a step-by-step outline, and the tasks are conducted consistently and appropriately. So many organizations have broad range of SOPs, and the technicians and the operators are trained to follow the individual SOPs. So tell me about the content of SOP. So whenever you have a question, you can answer like this: the objective or purpose or aim, scope, the responsibilities, procedure, precautions, annexure, abbreviation, and the references, revision history, etc. So why do we conduct stability studies? Friends, these are necessary for developing pharmaceutical products, and the influence of environmental factors like light, humidity, temperature on the API or the pharmaceutical ingredient to be evaluated. So expand BMR and BPR. Friends, BMR means batch manufacturing record, and these were prepared as a written file during the manufacturing a product by writing the step-by-step manufacturing process and details about the batch recorded here. So for every BMR, batch processing record, batch packaging record is held. So what is the difference between validation and calibration? Friends, validation will provide the written evidence so that the particular method or operation, so it is a written evidence. So you must highlight the point written evidence that a particular method or operation continuously develop a product with predetermined requirements and quality credits. So calibration denotes that equipment produces the values in a specific limits by comparing the values produced by a standard. It is done according to the calibration. So tell me any five countries with their regulatory authorities. So when you have been asked the question, you can simply start with India, Central Drug Standards Control Organization, next US Food and Drug Administration, next UK, which is the regulatory authority body of UK is Medicines and Healthcare Regulatory Agency, and for Japan, Ministry of Health, Labour and Welfare, and for Australia, which is simply TGA, Therapeutic Goods Administration. So what position of the oblong tablet to be placed in the hardness tester to know the hardness? Friends, this is a oblong tablet. It needs to be placed in lengthwise because the probability of breaking is more in this position. So what position of the oblong tablet? So we have already discussed this question in the previous slide. So why do we calibrate an instrument at a particular interval? So friends, calibration is very important for the equipment because there is a possibility for the instrument to drift out of the accuracy after qualification. So it needs to requalify the instrument at a specific time interval. So calibration is done at a particular intervals. So what checks shall be carried out during calibration of the disintegration test apparatus? So for disintegration apparatus, as you know, the disintegration apparatus move up and down in the basket. So the distance traveled, it shall be 53 to 57 mm. Temperature must be tested by using the probes and a standard thermometer. It shall be 37 plus or minus 1 degree Celsius. And the Stokes number per minute, it shall be 29 to 32 cycles per minute. So why is the positive pressure kept in the corridor, not in the process area? So this is very important question, friends, because the differential pressure gradient is generally essential at different locations to avoid cross contamination of the product through the air. So to prevent cross contamination of the product through the air. So differential pressure gradient is maintained. So recommended storage conditions for empty hard gelatin capsules. Friends, the recommended storage conditions are 15 to 25 degrees Celsius and 35 to 5% relative humidity. So what shall be the disintegration test temperature for dispersible tablets? Friends, 25 plus or minus 1 degree Celsius as per IP and 15 to 25 degrees Celsius per BP. So next question is, what is change request? This is similar to change control, we have already discussed. So difference between validation and calibration. This one also we have discussed earlier. So tell the time required for long-term and accelerated stability testing. Friends, accelerated stability means fast. So 6 months is enough. The time is 6 months. And for long-term stability studies, which is 12 months. So what is SISPQ? S means safety and I means integrity, S for strength, P for purity and Q for quality. So which fluorescence material is used in bin washing PQ? So riboflavin is used. Which class of area is required for tablets and capsule manufacturing? Friends, class D is required. So what is positive pressure? Friends, the atmospheric pressure is higher than the immediate surroundings area, usually measured in inches of water or pascal. So positive pressure is the atmospheric pressure which is higher than the immediate surrounding areas, measured in pascal. So what is Schedule M? Friends, Schedule M means good GMP, good manufacturing practices for premises of the plant, waste disposal and equipment. So it is two parts for factory premises GMP1 and GMP2 for plant and equipment. Schedule M1 and M2 like that. So what is deviation? Friends, deviation means deviating from the original. So deviation is an unexpected event that occurs during the ongoing operation at any stage of storage, manufacturing, analysis, and distribution of drug products. So the deviation is to be reported as and when events occur and must be investigated for the impact analysis. So what is VMP? Friends, VMP means Validation Master Plan. So it is about a brief information about the qualification, validation of and calibration of equipment, instrument and system. So validation provides the company's policy towards the validation program. Validation Master Plan will provide the company's policy towards the validation. So what are market complaints? Friends, it is an expression of dissatisfaction with a product or a service marketed. So what is a product recall? Friends, sometimes the product industry recalls the products back due to some reasons, which may be due to quality, safety or efficacy issue, which includes labeling, which violated the laws. So what is vendor qualification? Vendor qualification means the materials which are supplied by the vendor are according to the CGMP guidelines. So who is an approved vendor? So manufacturer of raw material, printed packaging material who are approved by the QA to supply the specific material from specific site based on CGMP. New vendor is nothing but formulation development or purchase department identifies a manufacturer supplying a specific material from a specific manufacturing site. So he is a new vendor. He's then if QA checks the vendor site, then begin procuring the material, then he is a approved vendor. So what is cleaning validation? Friends, cleaning validation means all the procedure used for cleaning are validated so that the equipment is suitable for processing the medicinal products. So annual product quality review. Friends, annual product quality review is a documented, regular, periodic or rolling quality review of all the licensed medicinal products to verify the consistency of the existing manufacturing process and to highlight any trends. To identify, we will know about trends, out of trends next in the next slide or another slide. Identify the product and process improvements or weakness for licensed medicinal products and appropriateness of the current specification for both starting and finished products. So what is the limit of individual unknown impurity in API as per ICH Q2A? So the limit is not more than 0.1%. Friends, what are class 1 solvents as per ICH Q3C guidelines? Friends, the class 1 solvents include benzene, carbon tetrachloride, 1,2-dichloroethane, 1,1-dichloroethene and 1,1,1-trichloroethane. Specific gravity of methylene chloride. The methylene chloride specific gravity is 1.3 gram per ml. And here is a question. Suppose the area is 100,000, then what are the maximum light and sound levels as per guidelines? You can skip this question. The light level is not less than 300 lux and sound level it is not more than 80 decibels. Friends, what is the instrument name which is used for measuring the vacuum in TARS? Macleod gas. So this is the instrument used for measuring the vacuum. So is the current good manufacturing practices requirements are only for the personnel in manufacturing? So it is a simple question because GMP current good manufacturing practices are for every department and every person of their specific area. So no, the answer is no. This requirement is for every employee of the organization who must know the relevant CGMP in his area. So why do we conduct the trainings? Friends, trainings are conducted to bring awareness, help employees become competent. So India belong to which climate zone? Friends, India comes under the climate zone 3, hot and dry zone, and climate zone 4B, hot and humid zone. So what is forced degradation or stress testing? Friends, forced degradation means testing the product at different temperatures and different environmental conditions to degrade the product forcibly by applying artificial methods. So explain about qualifications and their flow. Friends, these are the following qualifications which include design qualification, installational qualification, operational qualification and performance qualification. I recommend you to know about each qualification in detail. The flow include user requirement specifications, factory acceptance test, site acceptance test, DQ, IQ, OQ, and PQ. So what is a critical quality attribute? It is a physical, chemical, biological, and microbiological characteristics which should be under limits to ensure the quality of the product. So what is out of specifications? Friends, as a result which falls outside the established accepted specifications, then we can call an out of specification. So here you can see the example. So the specific limit for the assay is 90 to 110% weight by weight of the label claim, but the result is 85.2%. So then we call this situation as out of specification. So what is out of trend? Friends, suppose the results of a drug substance of a selected batch which is within the specification, and the similar batch of similar drug substance is falling outside the typical of the results of all the remaining batches. So this batch is different from the remaining batches, so it is out of trend. Example, here you can see the result obtained is 95.8% weight by weight. The results are within the specification. We should always then compare the result with the previous batch trend. If we found the typical value of the trend as 99, then this batch result is named out of trend. So the typical value is 99. So this is deviating, the result 95 is deviating from this, so it is out of trend. Friends, what is relative humidity? Relative humidity is the maximum amount of water vapor that air can possess. So if the temperature or the relative humidity of the area is going out of limit, so what action is to be taken? So we must immediately stop the line and inform the responsible department to raise the maintenance requisition slip. Ensure that all the intermediate materials are correctly covered. Next question is how to stop cross contamination in a area. Friends, by ensuring the proper gowning and material transfer procedure, controlled staff movement. So staff must move in the assigned lines or indications in the area, maintaining differential pressure, essential for controlling cross contamination. Friends, what is process validation? So there are different definitions. So we will read one definition. It is a documented evidence that the method which operated within the parameters established can perform effectively and reproducibly to provide a medicinal product meeting its predetermined specifications and quality attributes. So please read all the definitions. They may ask any of the definition. This is the EMA definition, European Medical Agency. So consistent with the regulatory guidelines, what are the stages of process validation? Friends, the stages of process validation are three stages which include process design, process qualification, and continued process verification. Here the next question is how many batches took for the process validation? Friends, generally a minimum of 3 consecutive batches must be taken with justification provided according to the EMA draft guidelines. And as per USFDA guidelines, the number of quantity of batches must be sufficient to supply the statistical confidence of method. So the number of batches taken must satisfy the statistical confidence of the method. So explain the strategy for process validation of solid dosage form. Friends, for validation of solid dosage form, we must use different lots. So the strategy is using different lots of raw materials like active ingredients, major excipient, and batches should be run in a serial and on different days and shifts. And batches should be manufactured within the equipment and the facilities designated for the commercial production. And the critical process variable should be set within their operating ranges, and failure to satisfy the wants of this protocol regarding the process should be subjected to process requalification. Friends, what is validation protocol? A protocol means nothing but a plan of action stating how process validation will be conducted. It will also specify the who is going to conduct the various tasks, and will specify the product and its characteristics. So what should be process validation protocol contain? Process validation protocol content, which include general information, the objective, background or pre-validation activities, and the list of equipment and their qualification status, facilities qualification, the process flowcharts, manufacturing procedure, list of critical process parameters, sampling tests and specifications, and acceptance criteria. Friends, the next question is what should be the blend sample size in the process validation studies? As per the studies, 1x to 3x dosage unit range on a case-to-case basis as per USFDA, and sampling sizes are often, normally it is 1 to 3x. As per USFDA, the sampling sizes are often increased from 1x to 10x with adequate scientific justification. Friends, how many sampling points should be considered for collecting blend samples? According to USFDA, at least 10 sampling locations are to be considered. So what will be the reason for within location variance of blend data? The inadequacy of the blend mix, sampling error or agglomeration are the reasons for within location variance of the blend data. Friends, during the process validation, why the Hopper challenge study is performed? This is done because the vibrations from the hopper influence the compression during compression may influence the blend uniformity. I mean, to evaluate the effect of for evaluating the effect of the vibrations during compression on blend uniformity, this hopper study is carried out. So, what are the critical process variables in coating? The critical variables are pan revolutions per minute, inlet and exhaust temperature, spray rate, gun distance, and air pressure. And why may the blend be critical parameter in tablet manufacturing? The answer is less blending will result in non-uniform distribution of the drug, and more blending will result in demixing. So proper blending is required. Friends, what is the mesh size of the dissolution test apparatus? It is 1.8 to 2.2 mm. So please remember this. Recommended temperature for dissolution testing, disintegration test for dispersible tablet. This is disintegration test apparatus mesh size, sorry. And 25 the recommended temperature for DT for dispersible tablets is 25 plus or minus 1 degree Celsius as per IP and 15 to 25 degrees Celsius as per BP. Friends, tell pass and fail test criteria for disintegration test. The goal is achieved. The test is passed if 16 out of 18 tablets disintegrate entirely. When the repeat the test on 12 additional tablets or units, if one or two fail to disintegrate, the goal will be achieved when 16 out of 18 tablets or capsule disintegrate entirely. Friends, these are the important frequently asked QA questions. I hope you like the information. Subscribe to our channel for getting regular job updates.